Hello viewers, let's learn about food microbiology today. All foods harbor one or more types of microorganisms except for a few sterile foods. Microbes have desirable role in food production, especially as fermented foods. Microbes can also cause food spoilage and foodborne diseases. In order to control such spoilage, understanding their role in food is necessary. To understand the role of microbes, they are isolated them as pure culture and characterized. Some of these simplest techniques for these studies were developed over the last 300 years. This episode deals with history and development of food microbiology, definition and scope of food microbiology, interrelationships of microbiology with other sciences, history and development of food microbiology. It is difficult to pinpoint the precise beginning of human awareness of the presence and role of microorganisms in food. The era prior to the establishment of bacteriology as a science may be designated as pre-scientific era. Pre-scientific era is divided into food gathering period and the food producing period. Spoilage of prepared foods apparently dates from 6000 BC. Western Europe started making pottery by about 5000 BC. The art of cereal cooking, brewing and food storage were either started at about this time or stimulated by this new development. Babylonian civilization were the first evidence of beer manufacture. Sumerians of about 3000 BC are believed to have been the first great livestock breeders and dairymen. They were the first to make butter, salted meats, fish, fat, hide, wheat and barley. Egyptians civilization shows use of milk, butter and cheese. The Jews were the first to use salt from the Dead Sea in the preservation of various foods between 3000 BC and 1200 BC. The Chinese and Greeks used salted fish in their diet, and the Greeks are credited with passing this practice to the Romans. Their diet included pickled meats. Mummification and preservation of foods were related technologies which has been influenced both Chinese and Greek development. Fermented sausages were prepared and consumed by the ancient Babylonians and the people of ancient China way back 1500 BC. The spontaneous generation of the life debate. The belief in the spontaneous generation of life from non-living matter was introduced by Aristotle, who lived around 350 BC. He concluded that aphid arises from the rainfall which falls on the plants and mice originates from the dirty food storage structures. Between the birth of Christ, AD 1100, few advances were made towards understanding the nature of food poisoning and food spoilage. Ergot poisoning caused by Claviceps purpurea, a fungus that grows on rye and other grains, is caused many deaths during the Middle Ages. During the AD 943, over 40,000 deaths occurred due to ergot poisoning in France, but it was not known that the toxin of this disease was produced by a fungus. During AD 1156, meat butchers came into existence. During 1248, Swiss were concerned with marketable and non-marketable meats. In 1276, a compulsory slaughter and inspection order was issued for public abattoirs in Augsburg. Until 13th century, the knowledge of relationship between quality and microorganism was not known. Kircher, a monk, in 1658 examined decaying bodies, meat, milk and other substances. He named the spoilage organisms as worms, invisible to the naked eyes. Kircher's description lacked precision, thus did not get wide acceptance. Anton van Leeuwenhoek, a tailor, used lenses to examine cloth. He discovered microorganisms, organisms so tiny that they were invisible to the naked eyes. He called these tiny living organisms animalcules. He first described bacteria and the protozoans. In 1765, Spelzeni showed that boiled beef broth, when sealed, remained sterile and did not spoil. Spelzenis performed this experiment to disprove the doctrine of the spontaneous generation of life. However, his work was criticized because they believed oxygen was excluded, and oxygen, which they thought was vital to spontaneous generation. During the same period, in 1795, a French confectioner named Nicolas Appert was granted patent for meat preservation when it was placed in glass bottles and boiled. This was the beginning of food preservation by canning. Today, this process is known as appertization. Nicolas Appert developed the first food preservation by canning. In 1837, Schwann showed that heated infusions remain sterile in the presence of air, which was supplied by passing it through heated coils. Critics concluded that heating changed the effect of air as it was needed for spontaneous generation. Louis Pasteur was the first person to prove the relationship between microorganisms in infusions and the chemical changes that took place in these infusions. Pasteur concluded that all the fermentative processes were caused by microorganisms. Fermentation, for example, alcoholic, lactic or butyric, were the result of specific types of microorganisms. He demonstrated the role of microorganisms in the spoilage of French wines, which resulted in rediscovery of bacteria. In 1857, he showed that souring milk was caused by microbes, and in 1860, he demonstrated that heat destroyed undesirable microbes in wine and beer. The later process is now called as pasteurization. Pasteur is known as the founder of food microbiology. He demonstrated that air needed heating to remain sterile. He finally disproved spontaneous generation. His experiment to disprove the old theory of spontaneous generation, microorganisms are the result of decaying matter, led to the new germ theory. It says microorganisms cause decay of matter and the microorganisms can be killed by applying heat, today known as pasteurization. In the 19th century, Industrial Revolution improved microscope. By 1838, Ehrenberg introduced the term bacteria and proposed at least 16 species in 4 genera. In 1875, Kohn developed the preliminary classification system of bacteria. Kohn was the first to discover that some bacteria produce spores, although, like bacteria, the existence of submicroscopic viruses was recognized in the mid-19th century. They were observed only after the invention of the electron microscope in the 1940s. The history of food microbiology is summarized in the table 1. Let's learn about development of history of food microbiology. It was in the year 1782, canning of vinegar was introduced by a Swedish chemist. Preservation of food by canning was patented by Appert in France in the year 1810. Peter Durand was issued a British patent to preserve food in glass pottery, tin or other metals or materials. The patent was later acquired by Hall, Gamble and Duncan, possibly from Appert. In the year 1813, Duncan, Hall and Gamble introduced the practice of post-processing incubation of canned foods. They also used sulfur dioxide as a meat preservative around this time. In 1825, patent preserving food in tin cans was done by Kensett and Daggett. Further, in 1835, patent making condensed milk was done by Newton in England. In the year 1840, fish and fruits were first canned. In the year 1842, patent freezing foods by immersion in an ice and salt brine was done by Benjamin in England. In the year 1843, sterilization was done by steam as a first attempt from Winslow in Maine. In the year 1853, patent for sterilization of food by autoclaving was done by Chevalier Apart. In the year 1854, Pasteur began wine investigations. Heating to remove undesirable organisms was introduced commercially in the year 1867 to 1868. In the year 1855, for the first time, powdered milk was produced at Wade in England. In the 1857, Louis Pasteur showed that souring milk was caused by microbes, and in 1860 he demonstrated that heat destroyed undesirable microbes in wine and beer. In the year 1865, the artificial freezing of fish on a commercial scale was begun in the United States. Eggs followed it in the year 1889. In the year 1874, the first extensive use of ice in transporting meat at sea was done. Steam pressure cookers or retorts were introduced at this time. In the year 1878, the first successful cargo of frozen meat went from Australia to England. In the year 1880, the pasteurization of milk began in Germany. In the year 1886, Krukowitz was the first to note the destructive effects of ozone on spoilage bacteria. In the year 1890, the commercial pasteurization of milk began in United States, and also mechanical refrigeration for fruit storage began in Chicago. In 1907, Metchnikoff and co-workers isolated and named one of the yogurt bacteria as Lactobacillus bulgaricus. The role of acetic acid bacteria in cider production was noted by Barker. In the year 1908, sodium benzoate was first time given official sanction by the United States as preservative in certain foods. In the year 1916, the quick freezing of foods was achieved in Germany by Planck, Ehrenbaum and Reiter. In the year 1917, Bird's Eye in the United States began work on the freezing of foods for the retail trade. Frank's was issued a patent for preserving fruits and vegetables under carbon dioxide. In the year 1920, Bigelow and St. published the first systematic study of spore heat resistance above 212 degree Fahrenheit. The general method for calculating thermal processes was published by Bigelow et al., and this method was further simplified by Ball in 1923. In the year 1922, St. and Mayer established method for studying Clostridium botulinum spores in phosphate buffer at 18 degree Fahrenheit. In the year 1928, the first commercial use of controlled atmosphere storage of apples was made in Europe, and it was first used in New York in the year 1940. In the year 1929, a patent used in France proposed the use of high energy radiation for the processing of foods. Frozen foods were placed in the retail markets, and a method was developed by Bird's Eye in the same year. In the year 1943, Procter in the United States was the first to employ the use of ionizing radiation, and also he used hamburger meat for preservation. In the year 1950, the D-value concept came into general use for the first time. In the year 1954, the antibiotic Nisin was patented in England for use in certain processed cheese to control clostridial defects. In the year 1955, sorbic acid was approved for use as a food preservative. The antibiotic chlorotetracycline was approved for use in fresh poultry, and oxytetracycline followed a year later, and this approval was reissued in the year 1966. In the year 1967, the first commercial facility designed to irradiate foods was planned and designed in the United States. The second became operational in the year 1992 in Florida. In the year 1988, Nissan accorded GRAS, that is the generally regarded as safe status in the United States. In the year 1990, irradiation of poultry approved in the United States. Let's learn about definition and scope of food microbiology. Microbiology is a specialized area of biology that deals with the study of microbes, ordinarily too small to be seen without magnification. Microorganisms are microscopic, and in Greek means micro means small and scopian means to see. They are independently living cells that live in communities. Microorganisms include large and diverse groups of microscopic organisms that exist as single cell or cell clusters. For example, bacteria, archaea, fungi, algae, protozoan and helminths, and the viruses, which are microscopic. Before the 1970s, food microbiology was regarded as applied sciences mainly involved in the microbiological quality control of food. Since then, the technology used in food production, processing, distribution and retailing and food consumption patterns have changed dramatically. These changes have introduced new problems that can no longer be solved by merely using applied knowledge. This discipline includes microbiological aspects of food spoilage and foodborne diseases and their effective control and bioprospecting of foods. It also includes basic information of microbial ecology, physiology, metabolism and genetics. This information helps to develop methods for rapid and effective detection of spoilage and pathogenic bacteria. It also is used to develop desirable microbial strains by recombinant DNA technology to produce fermented foods of better quality to develop thermostable enzymes. Let's learn about scope of food microbiology. This aspect is covered as bacteriology, mycology, parasitology, algae or phycology, virology, prions, epidemiology. Food microbiology in the changing world concerns international trade, consumer trends and communication, development of new technology, production of food ingredients. Let's now learn about interrelationship of microbiology with other sciences. In the early 20th century, studies continued to understand the association and importance of microorganisms, especially pathogenic bacteria in food. Specific methods were developed for their isolation and identification. The importance of sanitation in the handling of food to reduce contamination by microorganisms was recognized. Specific methods were studied to prevent growth as well as to destroy the spoilage and pathogenic bacteria. There was also some interest to isolate beneficial bacteria associated with food fermentation, especially dairy fermentation. However, after the 1950s, food microbiology entered a new era. Availability of basic information on the physiological, biochemical and biological characteristics of diverse types of food, microbial interactions in food environment and microbial physiology, biochemistry, genetics and immunology has helped open new frontiers in food microbiology. Among these are food fermentation or the probiotics, development of strains with desirable metabolic activities by genetic transfer among the strains, development of bacteriophage resistant lactic acid bacteria, metabolic engineering of strains for overproduction of desirable metabolites, development of methods to use lactic acid bacteria to deliver immunity proteins, sequencing genomes of important lactic acid bacteria and bacteriophages for better understanding of their characteristics. Food biopreservation with desirable bacteria and their antimicrobial metabolites, understanding of important characteristics of probiotic bacteria and development of desirable strains. Effective methods to produce starter cultures for direct use in food processing. Let's learn about food spoilage, identification and control of new spoilage bacteria associated with the current changes in food processing and preservation methods. Spoilage due to bacterial enzymes of frozen and refrigerated foods with extended shelf life. Development of molecular method, or we also call it as nanotechnology, to identify metabolites of spoilage bacteria and predict potential shelf life of foods. Importance of environmental stress on the resistance of spoilage bacteria to antimicrobial preservatives. Let's know about foodborne diseases, methods to detect emerging foodborne pathogenic bacteria from contaminated foods, application of molecular biology techniques and nanotechnology for rapid detection of pathogenic bacteria in food and environment. Effective detection and control methods of foodborne pathogenic viruses. Transmission potentials of prions from food animals to humans. Importance of environmental stress on the detection and destruction of pathogens. Factors associated with the increase in antibiotic resistant pathogens in food. Adherence of foodborne pathogens on food and equipment surfaces. Mechanisms of pathogenicity of foodborne pathogens. Effective methods for epidemiology study of foodborne diseases. Control of pathogen parasites in food. Let's learn about the miscellaneous aspects. Application of hazard analysis of critical control points or HACCP in food production, processing and preservation. Novel food processing technologies. Microbiology of unprocessed and low heat processed ready to eat foods. Microbial control of foods from farm to table or total quality management. To conclude, the human civilization began when hunters and gatherers adopted not only the production but also preservation of foods. Although the existence of microorganisms was recognized long back, its discovery was only in 16th century. Their importance on food spoilage and health hazard were conceived by our early ancestors. Ever since the association and importance in food were proven, efforts were made to understand the basic principles associated with food and microbial interactions. This knowledge was used to control undesirable microbes and effectively use the desirable types. Current investigations are directed towards understanding microbes at the molecular level. A food microbiologist should have a good understanding of the current developments in food microbiology as well as the characteristics of microorganisms important in food. Thank you, viewers.